For roughly 90 minutes after your last meal, your body is still managing the consequences of the first thing you ate. Not the last thing, the first. The sequence in which food arrives in the stomach determines the rate at which glucose enters the bloodstream. It determines the timing of the insulin response. It determines whether the incretin hormones, GLP-1, PYY, engage early enough to flatten the curve or late enough to chase it. In Alpana Shukla's 2015 controlled trial at Weill Cornell, reordering the same 628 kilocalorie meal, vegetables and protein first, carbohydrate after a 15-minute interval, reduced the 2-hour glucose area under the curve by 73%. The meal did not change. The biology of what happened next did. I did not have a name for what I was experiencing at the time. Most mornings by 10 o'clock, something had quietly collapsed. Not illness. Not anything a doctor would write down. Just the eyelids heavier than they should be. A concentration that had narrowed to a slit. A second hunger arriving 90 minutes after a breakfast that looked respectable. Oatmeal, some fruit, maybe toast. And the reflexive reach for a second coffee that helped for 20 minutes and then did not. I blamed the sleep. I blamed stress. I blamed the oatmeal. Then the fruit. Then carbohydrates in general. I was asking the wrong question. The mechanism was not the food. It was the order. The oatmeal first. The fruit first. The toast first. The carbohydrate landing in an empty stomach before anything slower had altered the digestive terrain. The evening version of the same mechanism has a different face. Dinner seemed moderate. Pasta or rice or bread first, because the bread is already on the table and the easiest thing to reach. An hour later, the appetite was not settled, but reopened, a pull toward something sweet, toward crackers, toward the refrigerator at 9 in the evening, with no conscious decision behind it. That pull is not a character failure. It is a glucose signal. The carbohydrate that landed first in the stomach produced a spike. The spike produced an insulin response. The insulin response pushed glucose below the fasting line long enough for the hypothalamus to interpret it as famine. The body responded the way bodies are designed to respond to famine. It called for more food. The meal the person believed had closed hunger had instead opened a second metabolic chapter. After 50, both of these experiences deepen. The body at 35 could absorb a starch-first breakfast with less drama. Fasting insulin was lower. Insulin sensitivity was higher. The pancreas responded faster and with less urgency. The body at 55 often cannot do the same. Fasting insulin rises with age. Central adiposity rises. Insulin sensitivity has drifted downward in the majority of adults who still believe they are eating the same way they always have. The same oatmeal and banana combination that once dissolved quietly into the morning now produces a glucose arc the aging beta cell notices. The collapse at 10 is not the oatmeal's fault. It is the order and the biology of the body it landed into. The scientific name for the experience is postprandial glucose excursion. The number that captures it across a 2-hour window is the incremental area under the curve, IAUC, total glucose exposure above fasting level, measured in milligrams per deciliter times minutes. When that number is large, the pancreas has worked harder than it needed to. The vasculature has been exposed to a spike it has to survive. And the subsequent insulin overshoot may push glucose low enough to restart hunger before the next meal was supposed to arrive. These are not abstractions. They are the 10 o'clock crash, the 9 o'clock refrigerator pull, named and owned. Alpana Shukla did not theorize her way to this finding. She fed the same 11 people the same meal twice. At Weill Cornell Medical College in New York, working within Louis Aron's Comprehensive Weight Control Center, she gave each participant, adults with metformin-treated type 2 diabetes, average age 54, BMI 32.9, diabetes duration 4.8 years, 628 kilocalories of ordinary Western food, ciabatta bread, orange juice, a chicken breast, lettuce, tomatoes, cucumber, vinaigrette. Same meal, same patients, 1 week apart. On one visit, carbohydrate came first, bread and juice, and 15 minutes later, chicken and salad. On the other visit, the order reversed. Chicken and salad first, bread and juice 15 minutes after. Blood glucose was drawn at 30, 60, and 120 minutes. Mean postprandial glucose was lower by 28.6% at 30 minutes, by 36.7% at 60 minutes, and by 16.8% at 120 minutes when protein and vegetables led. The incremental glucose area under the curve across the full 2 hours was 73% lower. Same pancreas, same meal, one variable, sequence. She extended the work in 2017 in BMJ Open Diabetes Research and Care with 16 adults with type 2 diabetes. Mean age 57.7. 3 conditions this time: carbohydrate first, carbohydrate last, or simultaneously as a sandwich. Carbohydrate last lowered incremental glucose area under the curve by 53% versus carbohydrate first, and lowered incremental insulin area under the curve from 9,769 to 7,354 microunits per milliliter times minute. A 25% reduction in the insulin demand placed on the pancreas for the same meal. In 2019, the group moved upstream into prediabetes. 15 participants, same meal design. Moving carbohydrate to the end attenuated incremental glucose peaks by more than 40%. 3 published trials, 2 metabolic populations, the same lever, tested repeatedly, holding each time. The physiology behind Shukla's numbers is not mysterious once you trace what happens inside the digestive tract in the first 20 minutes of a meal. Fiber-rich vegetables arriving first change the physical environment of the stomach before any starch reaches it. Soluble fiber thickens the gastric contents and slows the rate at which the stomach empties into the duodenum. This reduces the surface area available for rapid glucose absorption in the proximal small intestine. It is purely mechanical. The difference between pouring water through a dry funnel and pouring it through one already lined with a slow-moving gel. Protein does something the fiber cannot do alone. When protein reaches the duodenum and proximal small intestine, L cells in the intestinal lining detect it and release glucagon-like peptide 1, GLP-1, and peptide YY within minutes. GLP-1 does several things simultaneously. It stimulates glucose-dependent insulin secretion from pancreatic beta cells. It suppresses glucagon. It slows gastric emptying further, and it signals satiety to the hypothalamus. This is the incretin response, and its timing matters more than most people realize. When protein arrives first, GLP-1 is already circulating before the carbohydrate enters the gut. The insulin response is prepared. Pharmacologists call this first-phase insulin potentiation, meaning the pancreas receives an early signal and can secrete insulin faster and more efficiently when glucose finally arrives. When carbohydrate arrives first, glucose enters a gut that has not yet received the protein signal. The incretin machinery is slower to engage. The spike is steeper and longer before insulin catches up. The protein discussion established that GLP-1 and PYY are signals the gut already knows how to release. If the protein arrives before the starch, it has no reason not to. The gut is the pharmacy. Sequence is the prescription. A 2024 systematic review of meal sequence studies across multiple populations confirmed that vegetables, fruit, or protein consumed before carbohydrate consistently reduced postprandial glucose or glycemic variability, with mechanisms converging on delayed gastric emptying, reduced absorption rate, and enhanced GLP-1 and GIP secretion. Whey protein preload trials in type 2 diabetes found that a 50-gram whey preload reduces postprandial glucose by approximately 28%, while roughly doubling early-phase insulin secretion. A controlled mechanistic trial using a 30-gram protein and fiber snack bar taken 30 minutes before a meal 9.3 grams whey protein, 1.4 grams soy protein, 12.7 grams dietary fiber, 73 kilocalories total. Showed that taking the bar before the meal shifted the insulin curve to the left, raising early phase insulin and total GLP-1 compared with taking the same bar after. The bar added almost no calories. It changed the chronology of the hormone response. Same food, different hour. You now know why the first bite changes the hormone environment. The question changes from what the food is to what the digestive tract looks like when the carbohydrate arrives. Shigenobu Imai's group at Osaka University did not hand patients a continuous glucose monitor or a macronutrient spreadsheet. They handed them a sequence. 19 outpatients with type 2 diabetes, mean age 65.5 years, BMI 22.5, HbA1c 7.2%, plus 21 healthy controls, 8 mixed meals providing 17% protein, 25% fat, 58% carbohydrate, and 500 grams of vegetables daily, delivering 21 grams of dietary fiber. The instruction was simple enough to film. Eat the vegetables for 5 minutes first, then eat the main dish, the protein. Then, after a 10-minute interval, the rice or bread. On continuous glucose monitoring, the vegetable first sequence reduced standard deviation of glucose, mean amplitude of glycemic excursions, 1-hour breakfast glucose, and incremental glucose area under the curve at both lunch and dinner, in both the diabetic and the normal glucose tolerance participants. The finding did not require a metabolic diagnosis to appear. It appeared in people whose glucose regulation was considered normal. The vegetable first sequence changed what a healthy body did with an ordinary meal. The more consequential study ran for 24 months. Imai's group randomized 101 Japanese patients with type 2 diabetes to either a vegetables before carbohydrate instruction or a standard exchange-based meal plan. At 6, 9, 12, and 24 months, HbA1c had fallen further in the vegetables first group. Starting from comparable baselines near 8.3%, the vegetables first group reached 6.8% at 2 years. The exchange-based group reached 7.3%. That half-point difference, sustained and statistically significant across multiple time points, translates into measurable reductions in microvascular risk. And Imai's intervention did not require counting or weighing. It required reorganizing the plate. The cultural anchor here is not incidental. The behavioral pattern Imai's group formalized overlaps with something already present in traditional Japanese meal structures. Small dishes of vegetables, pickles, miso soup, grilled fish or tofu arriving before or alongside rice rather than after it. Rice was not always the first thing to land in an empty stomach. In traditional multi-course Japanese meals, the dense carbohydrate arrives relatively late, after the savory, the salty, the fibrous, the fermented. The glycemic break was already built into the choreography of the meal. Some cultures preserved it by habit. The laboratory confirmed what the habit had been doing for decades. You now know the sequence holds across 24 months in a real-world population. The question changes from whether it works to why the evening is a different problem than the morning. The same meal does not land the same way at 7 in the morning and at 7 in the evening. This is not a behavioral observation. It is molecular. In 2015, Christopher Morris, Kun Hu, Sally Garcia, and Frank Shear published a study in the Proceedings of the National Academy of Sciences using 14 healthy adults in an 8-day within-participant protocol. Identical mixed meals at 8 in the morning and 8 in the evening, under controlled laboratory conditions. Light, sleep schedules, posture, and physical activity all held constant to isolate circadian timing from behavioral confounders. Postprandial glucose was 17% higher in the biological evening than in the biological morning. Early phase postprandial insulin area under the curve was 27% lower in the evening. The pancreatic beta cell is not less responsive at night from fatigue. It is less responsive from circadian programming. The circadian discussion mapped why the same glucose load is handled differently at 8 in the morning and at 8 in the evening. The clock gene network in skeletal muscle and liver runs the processing equipment on a schedule, and evening is not when the equipment runs best. Peripheral insulin sensitivity is impaired by approximately 34% in the evening relative to the morning. Adipose tissue insulin sensitivity has been measured as 54% higher at noon than at midnight. Roughly 15% of skeletal muscle transcripts oscillate across the 24-hour cycle, including genes tied to glucose uptake, lipid oxidation, and mitochondrial function. The GLUT4 transporter, the primary mechanism by which insulin signals muscle cells to import glucose, follows a diurnal timing pattern, meaning the same insulin signal produces a weaker glucose uptake response in the evening because the downstream machinery is less primed to act. The morning terrain is forgiving, the evening is not. For the person over 50, this asymmetry compounds. Breakfast, arriving when insulin sensitivity is highest, GLP-1 response is stronger, and gastric emptying is faster, can tolerate a carbohydrate component after protein and fiber. The morning terrain handles imperfect execution. Evening demands a stricter version of the same rule. Dinner arrives when the beta cell is blunted, insulin sensitivity is depressed, and the circadian appetite, as Scheer's forced desynchrony work established, is near its daily peak. Eating refined carbohydrate first at dinner is the worst possible combination. The hungriest moment of the clock meeting the fastest absorbed food class at the moment of lowest metabolic efficiency. You now know the evening is metabolically a different terrain. The question changes from sequence alone to sequence plus timing. And what happens when dinner runs late? The appetite discussion showed that Scheer's forced desynchrony laboratory work placed the peak of subjective hunger at biological 8 in the evening, regardless of when the last meal occurred. In his 2013 paper in Obesity, Scheer and colleagues took 12 healthy non-obese adults into a 13-day forced desynchrony protocol. Sleep, meals, light posture, and activity scheduled across multiple circadian phases to isolate what the internal clock contributes to appetite, independent of behavioral cues. Hunger ratings showed a trough at biological 8 in the morning and a peak at biological 8 in the evening, with a 17% peak-to-trough amplitude. Appetite specifically for sweet, salty, and starchy foods oscillated by 14 to 25% across the circadian cycle, peaking in the biological evening. The reader standing at the refrigerator at 9 in the evening is not failing. They are being accurate about what their internal clock is signaling. The circadian drive toward evening eating evolved in an environment where food availability was uncertain, and caloric loading before the overnight fast made physiological sense. It does not make physiological sense in a body already carrying the metabolic residue of 3 meals, that the hour when glucose clearance is slowest. Then Chen-Juan Gu, Frank Scheer, and colleagues in 2020 made the late dinner penalty measurable with a protocol precise enough to be cinematic. 20 healthy volunteers, 10 male, 10 female, mean age 26, BMI 23.2, ate the same standardized dinner, either at 1800 or at 2200 hours. Sleep was fixed from 2300 to 700, regardless of dinner timing. Intravenous tubing threaded through a hole in the wall into an adjacent control room, so blood could be drawn hourly from 1700 until noon the following day, while participants rested undisturbed. A deuterium-labeled palmitate tracer mixed into a warm shake allowed the researchers to track exactly how much dietary fat was oxidized before morning. The late dinner shifted the postprandial glucose and insulin curves into the sleep window. 4-hour post-dinner glucose area under the curve was 522 milligrams per deciliter hours after the 2200 dinner versus 443 after the 1800 dinner. An 18% higher glucose exposure while asleep, with no behavioral variable changed except timing. Dietary fat oxidation was reduced, By the following morning, only 74.5% of the labeled palmitate had been oxidized after the late dinner, compared with 84.5% after the earlier one. Triglyceride peaks were delayed. Cortisol was elevated. Same food, later hour, different. For a 55-year-old whose metabolic flexibility, the ability to switch fluidly between glucose and fat oxidation, has already declined with age, eating late and eating starch first at that late meal is a compounding disadvantage. The glucose window narrows at night, fat clearance narrows, appetite peaks, all three converge at the same hour. You now know the evening compounds: blunted beta cell, depressed insulin sensitivity, peak appetite, slower fat oxidation if dinner is late. The question changes from why to what to actually do. Before the protocol, the evidence deserves its shelves. The established shelf holds the following with confidence. Acute meal order effects on postprandial glucose are replicated across type 2 diabetes, prediabetes, and healthy cohorts in multiple independent trials. The mechanisms: slowed gastric emptying, altered GLP-1 and GIP secretion, first-phase insulin potentiation, reduced proximal absorption rate, are understood at the cellular level and documented in humans. Circadian worsening of evening glucose tolerance is established, supported by controlled laboratory protocols and mechanistic data down to GLUT4 translocation timing and tissue-level gene oscillation. The plausible shelf holds appetite carryover from morning protein in adults over 50. The gut hormone changes are reliable. The downstream appetite suppression in older cohorts is real, but attenuated. Stein, Sonnen, and colleagues tested 30-gram and 70-gram whey preloads in 16 healthy older adults, mean age 72, and found meaningful shifts in insulin, CCK, GIP, GLP-1, and PYY. Yet Buffett energy intake was not reliably suppressed. In older adults, protein first is a powerful glycemic instrument and a decent satiety assist. It is not appetite anesthesia. The preliminary shelf is where any claim belongs that links meal reordering alone, independent of total diet quality, caloric load, weight change, and sleep timing, to reduced cardiovascular events or diabetes incidence. The physiology is directionally consistent. The long-term randomized evidence does not yet exist at scale. The claim this essay makes is narrower and more defensible. The first foods you eat restructure the glucose and insulin response to the meal. After 50, in a body where that response is already running with less reserve, restructuring it costs nothing and changes the experience of the same foods. A fasting glucose ordered at the annual physical. An oral glucose tolerance test with blood draws at 0, 60, and 120 minutes. A repeat HbA1c at 3-month intervals. A referral to a registered dietitian. A prescription for metformin starting at 500 milligrams twice daily and titrated upward. Possibly a DPP-4 inhibitor added when metformin alone is insufficient. Possibly a GLP-1 receptor agonist, semaglutide, tirzepatide, when the DPP-4 inhibitor is insufficient. A continuous glucose monitor worn on the upper arm, transmitting readings every 5 minutes to a smartphone. Possibly basal insulin at bedtime. Possibly prandial rapid-acting insulin before each meal. Cardiology and nephrology follow-up when years of postprandial hyperglycemia have deposited themselves in the vasculature. The cost of GLP-1 agonists currently runs from several hundred to over $1,000 per month before insurance. The cost of continuous glucose monitoring runs from $70 to several $100s monthly. The body's non-pharmaceutical version is one sentence: eat the vegetables and protein before the starch. That sentence is not a replacement for medication when medication is medically indicated. The honest dose comparison matters. The 30 to 73% reduction in postprandial glucose excursion from meal reordering is a meaningful acute effect. But a 2023 16-week prediabetes behavior change pilot produced modest real-world outcomes. Better vegetable and protein intake, roughly 3.6 pounds of weight loss, and only a trend toward HbA1c improvement. Meal sequencing is not Ozempic. It is the behavioral architecture that allows the gut's own incretins, the GLP-1 and PYY the L cells would have secreted anyway, to do more of their work earlier, before pharmacology or pathology has to do it for them. The stakes are not limited to how the morning feels. In the Nutrinet Santé cohort, Palomar Cros and colleagues followed 103,389 French adults, 79% women, for a median of 7.2 years, documenting 2,036 incident cardiovascular events across 699,547 person-years of follow-up. Meal timing was assessed through repeated 24-hour dietary recalls. Each additional hour of delay in the timing of the first meal of the day was associated with a 6% higher risk of overall cardiovascular disease. Each additional hour of delay in the last meal was associated with an 8% higher risk of cerebrovascular disease. Eating after 9 in the evening compared with before 8 was associated with a 28% higher cerebrovascular risk. Each additional hour of overnight fasting was associated with a 7% lower cerebrovascular risk. This is observational. Volunteer bias is present in large French nutrition cohorts. Residual confounding from shift work, light-at-night exposure, and sleep duration cannot be fully controlled. Causal inference is not warranted from this data alone. The glycemic variability discussion described why glucose spikes are oscillations the endothelium has to survive repeatedly. Frequency compounds the damage in ways a single HbA1c reading does not capture. The Nutrinet finding is the population signal. The Shukla and Morris data are the mechanism. Together they suggest that the first meal of the day and the last meal of the day are not trivial choices over time. They are, across years, part of the vascular exposure record the body is quietly accumulating. The morning protocol, the first 5 to 10 minutes of breakfast, belong to protein and fiber. Eggs and sautéed greens before toast. Greek yogurt with a handful of nuts before fruit. Cottage cheese or smoked salmon before oatmeal. Beans before a grain bowl. If juice appears, it appears after protein. If bread appears, it appears after something fibrous has already altered the gastric environment. The lab studies used 15-minute intervals between food groups, and that interval helped, but real life does not require a stopwatch. Sequence matters even without precision. The carbohydrate landing after something else is a meaningfully different event from the carbohydrate landing first. The evening protocol is the stricter version. Dinner before 8 when possible, not as a punishing rule, but as a metabolic preference. The GU and shear data are unambiguous. The same meal eaten 2 hours earlier produces measurably better glucose clearance and cleaner fat oxidation overnight. The meal opens with whatever is bulky, fibrous, and protein-rich: a salad, a vegetable soup, fish, chicken, tofu, legumes. Starch, rice, bread, pasta, wine, or dessert arrive later, in smaller portions or not at all, on evenings when dinner is late. The 10-minute structure Ima used in Osaka is practical and does not require a separate course. Vegetables and a protein while the rice finishes cooking, then rice. One contraindication that should not be buried: anyone using rapid-acting insulin analogs, Lispro, Aspart, Glulisine, or sulfonylureas that produce insulin independent of meal composition, should not quietly reduce or delay the carbohydrate portion of a meal without discussing the glucose curve change with their prescribing clinician. Changing when glucose arrives changes when the drug needs to act. That can narrow the hypoglycemia safety margin in ways the person will not anticipate from the outside. 3 in the afternoon finds me reaching for nuts and some leftover beans before anything else, even when the instinct is toward a piece of fruit. The instinct is not wrong. The order is. Week 1 typically produces less of the 10 o'clock energy collapse and less of the post-dinner appetite rebound. Week 4 tends to feel like steadier afternoon energy and fewer reflex snacks in the evening. By week 8, the same oatmeal, the same pasta, the same glass of wine no longer behave the same way. because those foods are now landing into a different hormonal environment than the one they occupied when they arrived first. The 10 o'clock collapse has a name now. The post-dinner refrigerator pull has a mechanism. The postprandial glucose excursion. The incretin window missed because the protein came second. The circadian beta cell blunting that makes dinner a different metabolic event than breakfast. The fat oxidation that slows when the meal runs late. The nutrient vascular signal accumulating hour by delayed hour across years. None of this required different food. It required different sequence. The same eggs, the same vegetables, the same rice. Reorganized by what lands first. The meal did not change. The first bite did.